The other case he wanted to look was the 10K proteomics sample, which is here. This is a much more complicated example because it has some huge numbers of clusters. You can see all the clusters up here. Most of them are very small. And let's again get rid of these axes. And so if you want to look at that, we try to position it right. We're on the best to try to, let's rotate it like this with the control shift operation. And now let's make it bigger with the, let's move it into the middle and make it bigger with the right mouse. So for this particular slightly, this is not totally square, this particular case, and so it's useful to orientate it so it fits in optimally on in your screen space. So you can see here sort of an interesting structure. There's some rather clear clusters with very close points very close to each other. Other clusters like these yellow ones here are much broader. Notice that there are a lot of clusters here, and then there are more clusters than there are discernible colors. And you can choose your clustering colors here. Here's one of the standard ones from MATLAB. That's one clustering, which seems to be what it was. If we use a Salzer custom, Salzer custom is just a somewhat different choice than MATLAB, which sometimes gives slightly better answers. MATLAB has 50 distinct clusters in it, and so it's the best in that regard. And now we can browse it, and we could try to look at this one here in more detail. So we have this clear, nice yellow cluster. Oh, it disappeared on me. Now let me center it on that. Press C. That moves it to the center. And now maybe you want to know what cluster that was. So to do that, we actually set the labels to be visible. And then we come over here and try to find out what the label here. And we can see that this is cluster 860. We look here, let's see what cluster it is. This is the point number and the cluster number and the label. And 860 hopefully is. Yeah, there's in this particular sample there are 248 points in 860. And so those yellow points are actually at this scale you can't really see it's a cluster, but it's obviously a cluster. Go to a scale like that. So that shows you how, and you see now actually I've made a mistake. I've flicked my mouse around and done a lot of mouse overs and picked up some extra labels. I probably didn't want to get rid of those labels. I just set labels to false. Then they disappear. So this is a rich data set, and one of the things you study on this, there's about 180,000 for the full sample, which we also have. And you can then study from this how well the experimentally determined clusters are actually together in this three-dimensional visualization. And remember, this comes from multi-dimensional scaling, where the whole point is the distance in 3 dimensions is meant to be a good approximation to the distance in the original space. So if things are close together in 3 dimensions, they're close together in the original space. If they're far apart in 3 dimensions, they're far apart in the original space. This is not done exactly because you can't exactly map from into the 3 dimensions, so these are approximates. But if you study our work on this field or other people's work, you'll find that actually it doesn't give a bad answer. So let's come back to our PowerPoint. So that tells you we've now gone through these three examples and used it to illustrate some of the basic principles of Plotviz. Let's now come to the next slides. So as an example of the synchronized camera view, let's go to an example which is given on this slide here. These are a couple of files in Plotviz format, which are actually the files we discussed in the X-Informatics class, with two different labeling into three groupings of a set of points in 3 dimensions. So let's go and do that. So to do this, we open one of them. Let's do this one, which is the one with the fanciest clustering. This one actually is a nice Plotviz file, so it actually has something else, which we'll discuss a little later on, called glyphs defined, which means we have special shapes. The larger glyphs here, the larger shapes are actually the cluster centers. And we're going to do a few things here. But most important, we're going to get rid of these measurable axes, which we don't like. And we also want to do something very important. Use sync camera to be true. So that's important. We have to go to camera, use sync camera and set that to be true. And then we go up to window and we tile the windows. Then we need to open. So and the file we're going to open is the actual original labeling for this particular data set. So there we have that. Before anything disastrous happens, and either we touch it and move it, we set that sync camera to be true for this. You have to do it, unfortunately, for every set you're looking at. We'll get rid of the axes again, and then we'll come up here and set tile windows. All right. Now, hopefully, we have 2 data sets. You can see here the one with original labels, and the other one corresponds to clustering with k-means. And now we can do anything we used to do before, but it will be done identically on these 2 data sets. So if we just sort of rotate it, you can see they're both rotating. And I say this particular original data set is not, well, does not have centers. Also, we need to do something else here, which I forgot to do. The glyph, because glyphs are computationally time consuming, they're defaulted to not properly correct if you rotate them. So you now, to make them look prettier, you set auto orientation to be true, and you have to do that for both data sets. Glyph auto true. And so now they look much better. That means as you rotate, it will automatically adjust the shape. So the shape also properly rotates. And so I can now actually switch and do the rotation here, and you'll find it follows on the other side. And there you are. That's an example of synchronized use within Plotviz. Very important actually in some cases. Even in some cases, the only way you can do certain things. So that's the end of that particular example. And that's why those files are there.